So till now we have seen two different types of nucleophilic substitution reaction, that is your unimolecular nucleophilic substitution as well as your bimolecular nucleophilic substitution. We also saw the various factors that affect the rate as well as the mechanism of your nucleophilic substitution reaction. Now we are going to proceed with another type of nucleophilic substitution reaction, which is called as internal nucleophilic substitution. Okay, so this is not exactly the same like your SN2 mechanism, but there is a difference in the mechanism which we'll be seeing in the further slides. So let's see what is internal nucleophilic substitution. Initially, when Walden was working with some of the alcohols, he observed that certain secondary alcohol in presence of thionyl chloride or phosgene is carbonyl chloride, COCl2, or phosphorus pentachloride, that is PCl5, in liquid SO3, liquid sulfur trioxide, or dry hydrogen bromide as the solvent at minus 80 degree gave a substituted halide. Okay, so the reaction had to proceed via your SN2 mechanism, and if SN2 mechanism is followed, then you very well know that the stereochemistry of the product that is formed will be inverted. Okay, suppose you start with a plus product, you should obtain a minus product. That is what we have seen for SN2 mechanism, that is the stereochemistry of the product gets inverted. Okay, but when he was working with plus malic acid, he observed that when this particular reaction is carried out in presence of thionyl chloride or phosgene or PCl5 in liquid SO3 or dry HBr, the product that is obtained is not with an inversion in configuration but with a retention in configuration. So here also substitution of your halide takes place, but the mechanism that this particular reaction follows shouldn't be the same as your SN2 mechanism. If it had been same, you should have observed your inversion product, but in this case the product that is obtained is not inverted product, but it undergoes substitution via retention of configuration at the stereo center. Okay, so why does that happen? Now we're going to see the mechanism of this reaction. So what happens in the first step, your thionyl chloride, that is SOCl2, attacks your hydroxyl oxygen. Okay, so the lone pair of electron moves towards your sulfur, and the pi electrons on your sulfur oxygen bond moves towards your oxygen. As a result, a bond between your sulfur and oxygen is formed, and oxygen, because it uses its lone pair of electrons to form a bond, it obtains a positive charge. Because this oxygen is attached to three bonds and only one lone pair is present, this oxygen becomes positive. Now, when this pair of electron gets back to its original place, then in order to satisfy its valency, this chloride ion leaves. Okay, so when this one leaves, it will abstract a proton from this hydroxyl group, and it will leave its electron over the oxygen, so that your oxygen gets back its lone pair of electrons, its shares of lone pair of electrons, and becomes neutral. Okay, so the attack of thionyl chloride gives you your chlorosulfite molecule. Now, this chlorosulfite molecule holds the nucleophile in a position which makes it feasible to attack from the same side at the reaction center. Okay, so what happens? This is held by a bond, and chloride again attacks this particular center from the very same side, and as a result, when the bonding is complete, this carbon oxygen bond breaks and oxygen takes its pair of electrons with itself. Okay, so when this bond is formed, your carbon oxygen bond breaks and it forms a much more stable SO2 molecule. So after the loss of this SO2 molecule, what you get is your substituted product. Okay, that is, when chlorine has to attack this, it has to make use of one of its lone pair of electrons, and since chlorine has used its lone pair of electron, it gains a positive charge, and in order to neutralize it, it has to somehow abstract this bonded pair of electron towards itself, and that is what it does. So your sulfur gets a positive charge, chloride becomes neutral, and since sulfur gets a positive charge, this negative charge on oxygen, that is delocalized towards your sulfur. As a result, S double bond O, S double bond O molecule is expelled, and this is a stable product. So after expelling this, you get back your product, the substituted product, which has the very same configuration as that of your starting material. Now, when they were working with these kind of compounds, they found that when a base was added to the very same reaction mixture, then again this reaction did not proceed with an retention in configuration, but it proceeded with an inversion in configuration, which means the mechanism that this particular reaction follows in presence of a base is not SNI now, but this becomes SN2. Then why is it so? What happens when you add a base? Why does the mechanism of the reaction change from SNI to SN2? Now we're going to see what happens. So, what would be the mechanism when a base is added to the very same reaction mixture? So, initially we saw that if we just add thionyl chloride in dry HBr or sulfur trioxide, liquid sulfur trioxide as the solvent, you should get a product with the retention of configuration. But when you add a base in this particular medium, what happens is initially your chlorosulfide is formed as in the previous case, like this. Okay, over here your hydroxyl oxygen first attacks your sulfur, forming a bond, and the whole sequence is repeated until you get a chlorosulfide molecule. So this is what happens over here also. So you can see after all that steps you get a chlorosulfide, and now your base comes into the picture. What does this base do? This base forms a coordinate bond with your sulfur. Okay, so when it forms a coordinate bond is by the complete transfer of electrons from one atom to the other. Okay, so the lone pair of electron is used for forming a coordinate bond, or you can say it is used for coordinating with your sulfur. Okay, so what happens? This lone pair of electrons moves towards your sulfur. As a result, the sulfur oxygen pi electron moves towards your oxygen, and then what happens? Your chloride is expelled. Okay, so here once your chloride is expelled, it leaves this molecule, and now it is free to attack from the either this side or this side. But when it attacks from the same side, it sees a steric repulsion because of the presence of such a big group. So the only possibility for the expelled chloride is now to attack from the opposite side, and hence it does attack from the opposite side, and that is why you get a product which is inverted. That is, the stereochemistry of the reaction center has become inversed. Okay, I hope it's clear now. So what you need to know is, when you treat this with thionyl chloride in presence of either HBr or liquid sulfur trioxide, you get a product which has retention in configuration. But when you add a base, then the product again follows SN2 mechanism with an inversion in configuration. Now, it has also been observed that certain solvents also affect the mechanism of this particular substitution. For example, if you take dioxin, Well, let me draw the structure of dioxin for you. It's something like, I have two oxygen. It's a cyclic heterocarbon. You have CH2, CH2, oxygen, CH2, CH2. So when in dioxin, if dioxin is used as a solvent, then retention in configuration is observed. While in toluene, you see that there is an inversion in configuration. This is ascribed to the ability of dioxane to coordinate with the carbon and assist the CS bond cleavage. So what happens, again the same thing happens as the mechanism of as you had seen in the mechanism when you had added a base like pyridine to your reaction mixture. Okay, so when malic acid is treated with thionyl chloride and pyridine, you get a product with inversion in configuration, whereas when the same reaction is carried out with an alcohol as the solvent, you get a retention in configuration. Likewise, if you use malic acid and if you use the solvent such as toluene, you get an inversion in configuration. If you take malic acid and you treat it with thionyl chloride with the solvent being dioxin, then you see that the product is retained. That is, it undergoes retention in retention of stereo center. So this follows your SNI mechanism. So these are some of the references from which all these materials were taken. You can just go through it and you can let me know if you have any other doubts. Thank you.